All right. Hello, everyone. My name is Raquel, and today I have the pleasure to introduce Professor John Cryan in our Brain Body Interaction virtual seminar series. I think you can hear us and see us well. All right. Let's see. Okay, perfect. Sorry, there's a little delay between the messages and when we can see it. So we can go ahead. I would just like to remind you that these seminars happen every Monday, and we bring together scientists which are interested in the interface between nervous system, metabolism, organ physiology, immunology. And so, yeah, stay tuned for updates in our website and in Worldwide Neuro Seminars. So before introducing our wonderful speaker, let me remind you that you can ask any questions during the talk. You can use the Ask a Question button. And also, if you want to meet the speaker after the talk during a Zoom Q&A session that we have, if you haven't registered for it yet, please send us an email. I will write the email address in the chat in a bit. So now for real, today we have Dr. John Cryan, who is a professor and chair of the Department of Anatomy and Neuroscience at the University College Cork. He received his bachelor's in science and PhD from the National University of Ireland. He was a visiting fellow at the Department of Psychiatry at the University of Melbourne in Australia, which was followed by a postdoctoral fellowship in the United States at the University of Pennsylvania and the Scripps Research Institute in California. He spent 4 years at the Novartis Institute for Biomedical Research in Basel, Switzerland, as a lab head prior to joining the University College Cork in 2005. He's a PI also in the APC Microbiome Institute. He has published over 250 peer-reviewed articles and book chapters, and he, as a member of the highly cited researcher list, was also included in the 2014 of the world's most influential scientist minds. So it's a real pleasure that he is with us today. He is in the editorial board of several journals and have received many prizes that I'm not going to start listing now. We want some time to listen to him. And today he will be talking about the microbiome gut-brain axis across lifespan. So with this, I finally leave the stage to Dr. Cryan.
Thank you very much, Raquel. Thank you for inviting me here to this wonderful symposium series. It's really great to be here. So today, yes, I'm going to share with you the adventures that we've been on in Cork, here in the very south of Ireland, for well over a decade now, implicating what's going on in the gut with what's going on in the brain. Overall. So, you know, it's really about getting to the biology of gut feelings overall. So here is a very famous painting from Matisse called the Bateau, and it hung as part of a retrospective exhibition in the Museum of Modern Art in 1961, and it hung like this, until a very eagle-eyed stockbroker called Genevieve Hebert noticed that something was wrong with this painting. And, you know, thousands of people had passed by, including Matisse's son, and what they had not noticed was that people were looking at the painting.
Sorry, Jen, I have to interrupt you. I think people are seeing your screen really small. I don't know if there's any possibility to try the other full mode presentation.
Oh, yeah, sure. Let me just see my screen.
Sorry, guys, we had some technical difficulties before.
How is it now, Rebecca? I have to share now. Share screen. application window. Oh, and then I have to make this bigger. Okay, that's what's wrong. How is it now?
I can definitely see it bigger. I hope people in the audience as well.
Hopefully.
Yes, I think so. So we're good to go.
So anyway, you get to see this wonderful painting from Matisse a little bit longer. And so it was hanging like this, and then Genevieve Hebert noticed that something was wrong with it. And what was wrong with it, it was that it was being hanging upside down and needed to be turned the other way around. And so the take-home message from this is that sometimes we have to look at things a little bit differently to be able to understand how they really work. And as a neuroscientist, this is really what I try to do, is really look at things upside down in a different way. And maybe by doing that, we can see how things should really be. So I work in a medical school, like many of you, and in medicine, almost everything goes back to Hippocrates. And Hippocrates is thought to have said, now it could be fake news, but he's thought to have said that all diseases begin in the gut. But when I talk to my psychiatry or my neurology friends, they, might question this. And so hopefully I can convince you that there is some underlying biology to this. And all we have to do is look to our language. We use phrases like gut feelings and gut instincts. We make gutsy moves when we're brave. We're gutted when we're disappointed. We go with our gut. We have butterflies in our tummy if we're nervous. So perhaps, the underlying biology of these emotions could be really important. So more recently, a new player has really emerged in terms of our understanding of gut-brain signaling, and that is the microbiome. And so there are a few general things that I want to remind people about the microbiome. First of all, we are living in a microbial world. Microbes were here long before humans, and it's important to think about this in an evolutionary context. In terms of numbers, it is really humbling to think that when we look at the genes that we have in and on our body, we are more than 99% microbial. It's also humbling as a neuroscientist to think that the weight of our gut microbes is roughly about the same as that of our brain. And indeed, this is a very fast-moving field. And if I'd given this talk some years ago, I would have told you we have 10 times more microbial cells than we do human cells. But this has been revised downward. So we're still more microbial, but just about. So about 1.3 to 1 by current estimates. So just think the next time you go to the bathroom and you get rid of some of your microbes, that you're becoming more human and what that might mean. When we talk about the microbiome, for the most part, actually, we don't mean the microbiome, we mean the bacteriome, because that's really what we're looking at. And but our microbiome is much more complex, involving our archaea, our viruses and bacteriophage, our fungi, protists, nematodes. So it's a much richer ecosystem, and we're only slowly coming to grips with the complexity of this ecosystem. It has made the cover of all the glamour magazines, as you can see here, and the lay press love it overall. And one of the things I wanted just to reinforce, and this was part of a framework document that we did some years ago, led out by Seth Bordenstein and Kevin Thays, was to really make sure that we put the microbiome within a proper eco-evolutionary framework. And to show that humans are a complex mixture of both their genes and their microbiome, and that this microbiome is shaped by what's going on in the environment overall. And so the concept of a hologenome, all of our genes and our microbial genes, has gained a lot of attention over the past years. And what we're really trying to do in my lab is trying to understand how microbes can talk to the brain and influence behavior. And so these mechanistic challenges are one of the big things that we're focusing on right now. And the second one, and I'll touch on some of this towards the end of the talk, is how do we move from all of these amazing studies in animal models all the way to the human condition. And what we're paying a lot of our attention on right now is focusing on the relationship between the microbiome and the brain at critical windows across the lifespan. In early life, in adolescence, and all the way to aging. Where does it begin? Well, for the most part, we're taught to be sterile in utero. And we get our microbes kind of like a birthday present from our mothers on the day we're born as we emerge through the birth canal. And these early microbes, frontier bugs, can start to inform the developing immune system. They can start shaping other systems within the body. And so, but it is that first 2 years of life that's critical in laying down these microbes. And so factors that can influence that can have play a role in altering the trajectories of this programming that affects most systems within the body. Factors including diet, whether you're breastfed or bottle-fed. Host genetics and epigenetics are very important. The environment you live in, exposure to medications like antibiotics, and various other lifestyle factors. And one of the factors that we've been interested in has been mode of delivery at birth. Because this goes back to work from Gloria Domingos-Bello and colleagues. They showed well over a decade ago now in a kind of pilot study that infants born by C-section have a very different microbiome than those born per vagina. And so basically in this evolutionary relay race of mother handing over microbes at birth to the infant, in C-section it kind of just stops the handover. And so this has been replicated in many studies. The most notable is the TEDdy study, which had over 12,000 subjects, and it showed very clearly that birth mode was associated with a different microbiome, and the cover of Nature covered it as kind of a altered security blanket and changing the security blanket. Moreover, work from Trevor Lawley's group has shown that it's not only the missing microbes that are playing a role, but it's also the blooming of pathogenic bacteria that occurs during this critical window. And so we know that being born by C-section increases the relative risk of asthma, allergy, type 1 diabetes, all of these immune-related disorders. But we know very little about it in the context of brain health. And this is something that we've been studying in our lab. And to study it, we had to develop an animal model. And this is work from Livia Mores, who's now a postdoc in Caltech with Sarkis Mazmanian. And Livia, we developed a really nice mouse model of C-section. And what we found was that these animals had very early in life deficits in the levels of bifidobacteria in their microbiome. And these deficits were coincident with alterations in behavior as early as postnatal day 9, but that these changes in behavior persisted into adulthood, most notably social behavior and anxiety-related behavior. And then we sought to see, could we rescue these changes? Could we do something about it? And the first thing we did was to take advantage of the fact that mice are coprophagic. They eat each other's poo. And so we use this as a kind of an artificial fecal transplant, if you would. And so by co-housing mice that were born by C-section with mice that were born naturally, we were able then to see, could we rescue the microbiome deficits? And we could. And then we also were able to rescue some of the behavioral effects. We also treated these mice with a probiotic that was, in this case, it was a bifidobacteria itself, or a prebiotic that stimulates the beneficial bacteria, the bifidobacteria in particular. And both of these rescued these deficits. You know, so these are really interesting findings to show that there are enduring effects of early life dysbiosis that continue all the way into adulthood. And we've done similar studies with antibiotic treatments, but really to get at, to actually convince my very skeptical neuroscience colleagues that the microbiome in early life could be shaping brain and behavior, then we need to have more evidence. And the best way to get evidence that something is important, we can learn from engineering or other aspects of biology, is to take it out and see what happens. And so we've been working a lot with germ-free mice, and they've allowed us to show, and other groups, most notably the groups of Rochelle Heitz and the Karlinska and Jane Foster in Canada, but many other groups have shown that there are changes in brain and behavior in these germ-free mice. One of the key findings we made some years ago was that there was a change in the myelination pattern in the prefrontal cortex of germ-free animals. That was quite surprising because it was very specific to the prefrontal cortex, and it was very specific to male animals. And when we went down the electron microscope, we could see that there was a hypermyelination occurring, which was quite interesting overall. Moreover, we then wanted to look and see what happens at the level of the hippocampus. We're very interested in the hippocampus in the context of higher cognitive function and in terms of adult hippocampal neurogenesis. And again, you can see these are new neurons in yellow here that are being formed in the dentate gyrus of the hippocampus in adulthood. And in the absence of microbial signals, the mechanisms underpinning the regulation of this neurogenesis has gone awry. Other changes we have is in the relationship to neurotransmitters, most notably GABA and serotonin, but also in relation to genes involved in plasticity, BDNF and other trophic support genes, and also in dendritic growth. Microglia work from Marco Prince's group and Sonia Gorel have shown that microglia are not functioning appropriately in germ-free animals, and then work from Karlinska showed that the blood-brain barrier, from Sven Patterson's group, showed the blood-brain barrier integrity was disrupted in germ-free animals. Here you can see this is a basolateral amygdala neuron from one of our studies some years ago, showing that there is a increased arborization occurring. It's almost like this amygdala neurons in a chronically stressed state already. And we know the amygdala is very important brain area for fear learning, for pain processing, and for social behavior overall. More recently, and this is work just published from Henry Dark, a postdoc in the lab, together with Michael Collins and Ken O'Riordan in the group, that they were interested in hippocampal long-term potentiation, and using the multi-electrode array system, we're able to show very clear deficits in long-term potentiation, in the molecular basis of memory, if you will, in animals that were born germ-free, but only in male animals. Female animals were able to mount LTP pretty well. So this is quite intriguing. When these sex-dependent effects, we're beginning to see them more and more common, and there's something about the male brain that makes it susceptible to deficits in the microbiome, or there's something about the female brain that makes it quite protective. At the behavioral level, then, based on following on from the amygdala deficits we saw, we also see changes in amygdala-dependent fear learning, as you would predict, and amygdala-dependent visceral pain processing, and in amygdala-dependent social behavior. And here we can see that mice, while they're a bit like humans, they're quite social animals. So if you give them the chance to spend time with another mouse or an empty chamber, they'll gravitate towards the mouse, but not if they're germ-free. Mice, perhaps like some humans, can be a bit fickle. So if you give them the chance to spend time with a new playmate versus their everyday playmate, they'll gravitate towards the new playmate. But again, not if they're germ-free. These mice also had increases in repetitive behavior. So what we're seeing is some of the core symptoms that are disrupted in disorders like autism and schizophrenia are also gone awry in our animals that don't have microbes in our gut. Or you could frame it in a different way, that you need appropriate microbes in your gut to have normal social behavior if you're a mouse. So we've been digging into some of the molecular mechanisms that are at play. So by just, forcing social interaction in mice and seeing what pathways in the amygdala get activated, we show, for example, in normal circumstances, you get an enrichment of genes involved in the MAP kinase signaling pathway, but this is not happening in germ-free animals, but instead the enrichment is happening at the level of the spliceosome and RNA splicing events in particular. Now, this is just scratching the surface of mechanism, but it's hinting that of some of the processes that are gone awry that might be leading to some of these social deficits. An interesting work from Dan Geschwin's lab has shown that there is widespread RNA editing dysregulation in postmortem tissue from individuals with autism overall. And so what we're beginning to see, and this was work that Roman Stilling did when he was a postdoc in the lab, we started thinking about okay, so we need to have microbes for normal social interaction. So from an evolutionary perspective, you kind of start asking questions like, what's in it for the microbes? And so we collaborated with Seth Bordenstein at Vanderbilt and put some of these ideas to Seth. And Seth is an evolutionary microbiologist. And Seth basically told us in a very nice way that we were completely wrong to even be asking these questions in the first place. And the reason we were wrong was that I had forgotten something that I've already told you guys, and that is that the microbes were there first. And so there never has been a time where the brain has existed without these microbial signals. And we have co-evolved with these friends, and these are our friends with benefits, and perhaps they're our friends with social benefits. And so that's really an important aspect. And when you start thinking about brain microbiome interactions in an evolutionary context, you start to looking at it in a different way. And then more recently, we've gone and looked at this across the animal kingdom. And what you see is if you start interfering with the microbiome across different species, whether it's in insects all the way to non-human primates, you interfere with social hierarchies and social behaviors, like we had shown in mice. And the flip side of it is also true, that if you start interfering with social behavior and social dominance, then you start interfering with the composition of the microbiome. So during our lockdowns, and as we're talking in Ireland now about wave 4 of this pandemic, the question is, what are all of these lockdowns doing to our microbiome? And if they are doing anything, could it be negatively affecting how our brain gut microbiome axis is operating? It's all conjecture right now, but it's still very provocative to start thinking about things in this holistic way. And it underpins where we want to go with this. And so that we're beginning to see that, the microbiome gut-brain axis is really a social brain axis, and that the core circuits underlying social behaviors are being influenced by what's going on in the microbiome. And we can influence this by a variety of different ways, most notably by diet. And the questions then remain whether any of the influencing factors that we have on the microbiome and then, in terms of social interactions and on this microbiome gut social brain axis, can they also influence social behaviors? Whether, can we target the microbiome to influence how people show empathy or cooperation or teamwork or altruism or trust or decision making? All of these things. This is quite provocative, and it's something that, along with my collaborators, we're beginning to start to study now more and more. But again, it shows that the relationships between the social brain and the microbiome, and we've a long way to try and understand this. So what we're beginning to see is early in life, we know, going back to Hubel and Wiesel, that there's critical windows in brain development and brain plasticity that give rise to various higher order neurocognitive functions. The question remains, what are the plasticity and critical windows in the microbiome? And if we juxtapose these together, where do we go? And trying to understand the relationship of the microbiome and informing executive function, for example, early in life, in informing social behavior and informing normal aspects of cognitive function and cognitive development. We're also beginning to see, if we start looking at the Barker hypothesis and the developmental origins of disease, where we really take into account both the prenatal and postnatal environments, we know that the microbiome could be another pillar in this, along with host genetics, in influencing what's going on early in life to later brain development and later brain function overall. But we have a long way to go to try and tease that apart. So that's early in life. Next, I want to move to adolescence. And the adolescent brain is something that I've been really interested in for a long time. And I'm the professor of anatomy in the university here, so it's worth pointing out, and any of you who have teenagers will know this, that the anatomy of a teenager's brain is very different to that of a young or adult brain. And really what we're seeing is that it's a brain that's very much under construction. And it's one of the hottest areas in all biological psychiatry right now, understanding how synaptic pruning events that happen during adolescence can lead to long-lasting effects, and understanding microglia neuron interactions and how they're shaping brain function into adulthood, and how this can give rise to a variety of different disorders, and particularly psychiatric illnesses, ranging from impulse control disorders to adolescent onset schizophrenia. And a lot of focus in the field has been on insults like alcohol, stress, poor nutrition, drugs of abuse, inadequate sleep, all of which can influence the developing adolescent brain. But equally, they can also influence the developing adolescent microbiome. And this has received much less attention. And this may also play a role in a variety of these disorders. And I mentioned some of them already, but also shout out to eating disorders as well as substance abuse disorders. So this is something worth noting. And it's something that we've been interested in some time in studying. For example, this is work from Christine Fulling in the lab, showing how deleterious diets can have negative effects on the microbiome. And then we can also look at whether positive diets could affect or have mitigating effects on some of the negative effects of stress on the microbiome during adolescence. And this is work in collaboration with Beatrice Pisani's group, where we showed a diet enriched in vitamin A and polyunsaturated fatty acids was able to reverse the stress-induced deficits in the microbiome during adolescence. We've also been studying the impact of antibiotics. Earlier work from Leva Debonnet in the group was showing that there are long-term effects of adolescent manipulations of the microbiome. And we more recently followed up with Gilly Lack's work, where we were able to show that even when you stop taking the antibiotics, there were enduring effects on microbiome composition way into adulthood. So we're beginning to see the adolescent microbiome as a critical period for studying gut microbiome brain interactions overall. Then I want to move quickly then into aging. Here is Eli Metchnikoff. Now, I guess most of you are neuroscientists or people interested in metabolism, so you may not know who Metchnikoff is. And he's like one of the heroes in the whole microbiome field, an unlikely one. He was a Russian working at the Pasteur Institute in Paris, and he won the Nobel Prize in 1908 for his really important work on the discovery of phagocytosis, how the immune system gobbles up foreign bodies. And, well, as often happens, great scientists, later on in their careers, they can start coming up with crazy ideas. And Metchnikoff was full of crazy ideas. And some of them were complete nonsense. But one of the ideas that he had and was really fixated about was, why did people in what's now part of rural Bulgaria, why did they live longer and healthier? And what he put it down to was what they ate. And what they ate was a lot of fermented foods containing lactic acid bacteria. So he's not only a father of immunology, he's a father of geriatric science. He's also a father of probiotics. And if you go to Korea, for example, you can get a yogurt drink with his picture on it. So it must have that Nobel sign of approval and must be good for you, of course. But anyway, most of what Metchnikoff studied was ignored in the context of microbiome healthy aging for many years. And it's really important to really realize that aging has, all aspects of aging have now been implicating the microbiome in mediating their effects. Effects on the immune system, immunosenescence in particular, and cardiovascular system, the gut, and even on the brain. And work from my colleagues here in Cork showed almost a decade ago now that the composition of the microbiome and aging was very important factor in driving health outcomes, in particular frailty. And the factor that was driving the compositional changes in the microbiome level was diet. And it was all down to diversity in the diet. And just like in all aspects of life, diversity is really key in the aging microbiome. And the more diverse our diet was, the more diverse our microbiome was, the better the health outcome. So we've been really interested in this overall, and we've been revisiting the concepts of Metchnikoff and showing that there are age-related changes at the level of the immune system, at the level of behavior, at the level of the microbiome in aged animals. And the next part of our puzzle was, could we do anything about it? Could we reverse some of the effects of aging by targeting the microbiome? And for this, we decided to focus on middle age. It's a time period I'm very interested in right now for obvious reasons. And so the question here was, if we supplemented the diet with a prebiotic, in this case an inulin, which stimulates the growth of beneficial bacteria, could we counteract some of the effects of aging? And what we focused on in particular, and this is work led by Marcus Boehm, together with my colleague Harold Shellikens and Ted Dinan, we showed that inulin, which is very much heavily present in leeks and onions and artichokes, could reverse the neuroinflammatory effects of middle age in terms of stress-induced immune priming, the infiltration of inflammatory monocytes, increased microglia activation. We see all of this in middle age, as you can see here, but when animals were given this inulin, it blunted all of these effects. And so my take-home message for these studies is that if you're thinking of having a midlife crisis, maybe forget about the motorbike and turn to the leeks instead, and it might be better overall. We're also now looking at other ways, and so we have really exciting new data looking at the ability of basically doing a fecal transplant and transplanting from young animals to aged animals to see if we can reverse some of the effects and kind of do it in a kind of fecal parabiosis way. And the data on that is emerging and quite exciting as well overall. But irrespective of what period across the lifespan we're interested in, the big conundrum in our field is how do microbes in the lumen of the gut send signals to the brain and influence key circuits underpinning behavior? And we're just really beginning to tease this apart. And one of the things that's very clear is that the immune system plays a key role, that the integrity of the gut barrier is very important, that the second brain, the enteric nervous system, is very important in mediating these effects. And here also we have the vagus nerve, and this has been a pathway that we've been particularly interested in our lab for well over a decade now. And indeed, in a paper published 10 years ago, which is kind of one of our big papers, early on papers, together with John Bienenstock's group in Canada, we showed that when you cut the vagus nerve, all of the effects of a specific lactobacillus or Lactobacillus rhamnosus were completely absent. These were effects on the HPA axis, effects on behavior, and effects on neurotransmitters were completely gone when we cut the vagus. So what this means is that what happens in vagus doesn't just stay in vagus, but will actually affect our emotions. And we're, really trying to use this, and work Christine Fulling has been doing in the lab is trying to understand the pathways of communication from vagus into the nucleus tractus solitarius, and then onto key circuits underpinning behavior, and applying state-of-the-art neuroscience tools to dissect out these circuits. And that's ongoing work that we have, and we're very excited about in the lab overall. The other thing to remind you is that the microbiome is like a factory, producing all kinds of weird and wonderful chemicals that our bodies wouldn't be able to produce without it. And among these are the short-chain fatty acids. So this was really important for us because it led us to the idea of, could we just bypass the microbes and go to the short-chain fatty acids themselves? And that's kind of like a postbiotic approach. And in this paper, we showed that indeed, and this is work from Marcel van der Waal when he was a PhD student in the group, he showed that we could attenuate some of the selective effects of chronic stress, both at the level of the gut and behavior, when we pre-treated with a cocktail of short-chain fatty acids substances. So as a pharmacologist by training, this is a really nice approach. And the questions we have in the field is, what are the other, short-chain fatty acids are always the low-hanging fruit. What are the other metabolites? And what are their molecular targets? And can we understand what they are and deorphanize them and try and understand how the microbiome could be targeting host receptors in a way that will drive changes in brain and behavior overall? So this brings us to the concept of psychobiotics. This is a term that we coined here in Cork together with my colleague Ted Dinan. And psychobiotics, we've expanded its meaning. They're basically ways of targeting the microbiome for mental health benefits. There are different approaches. We've mentioned some of them already. Probiotics, fermented foods, prebiotics, postbiotics like the short-chain fatty acids. Paraprobiotics is an interesting concept. Maybe the bacteria don't need to be alive. Maybe it's what's decorating their cell wall and how that interacts with the host immune system that's really important. Or fragments that come from them. Symbiotics then are combinations of probiotics with prebiotics. And so this is something that we're really excited about. There are different approaches that can be used at different phases across the lifespans in different conditions overall. We wrote this book, The Psychobiotic Revolution. It's a bit hyperbolic to say revolution, but it's beginning of a new way of thinking, a new way of understanding how we may manage our brain and our body and our mental health and well-being by targeting what's going on in our microbiome. It's early days. It's about communicating the importance that the microbiome has for all aspects of our health. And we, as I said, I work in a medical school, and we have medical training where our dentists get more nutritional advice than our actual physicians. So we need to do more in trying to inform the public and inform people the importance that diet and the microbiome and nutrition has in overall health in general. Now, do we have any data? Well, I've shared some already, and this is kind of a summary slide showing that a variety of mechanisms, probiotics, prebiotics, postbiotics, all can have beneficial effects in terms of modulating behavior. What's really important to notice that there's a molecular underpinning to this at the level of neurotransmitters and neuromodulators that are slowly being unraveled. But I will also want to highlight that for the most part, most manipulations of probiotics in particular won't do anything. They probably won't do any harm, but they won't have any benefit. And understanding why specific strains have specific effects is going to be an important part for the field moving forward. I also include here milk kefir, because we just published on that last year. Kefir is a fermented food, and this is really exciting work, and it's something that I'm quite excited about, because it's something that everyone can do in their own lives, is take kefir from milk, and it doesn't cost anything more than the actual milk. And some of the effects we found are really, really, really important. And we have more data brewing on that together with my collaborator, Paul Cotter, here at Teagasc and the APC Microbiome Ireland. In terms of translating these animal studies to humans, well, we have some beneficial effects already. This is a small study we did some years ago. Here's Andrew Allen, the psychologist in our team who led this, where we took healthy volunteers and we brought them into the lab, and we have very nice ethical ways of stressing people in the lab. In this case, they have to submerge their arm in a ice bucket of water, and there's someone looking at them, taking notes and judging them. So there's a psychological component as well as a physiological component to the stressor. And we found that those that had taken this same bifidobacteria that we had shown to have beneficial effects in animals, also that they didn't have the same stress response. It also had pro-cognitive effects and also changed brain EEG. So this was really, really remarkable for me to see something that had come to our animal studies being translating to humans. Paul Eng's group in Tübingen they came to us and were looking for a candidate strain of bacteria to test in their imaging studies. And so this is a human brain imaging study where they performed a cyberball task in the scanner. There's a person in the scanner playing a game with two other people, and they're passing a ball from one to the other. And all of a sudden, the two other people start excluding the person in the scanner. And your brain just doesn't like to be excluded. It takes you back to all these playground taunting or whatever. So the brain mounts a stress response. And if they'd taken this bifidobacteria, the stress was greatly reduced. So that's really neat. And we were very excited about these findings. Again, validation of our animals, going from animals all the way to humans. And so then we took our most potent psychobiotic from our animal studies and tested it in a similar way in humans. And you cannot get more negative data into the one paper if you tried. And boy, did we try. I mean, it really highlights the difficulties we have in this field, but it's not specific to the probiotic field or the microbiome field. It's in all aspects of behavioral science in general, in trying to translate healthy volunteer studies in animals all the way to humans, and then also to patients. But what we're beginning to see, and it's beginning to emerge more, is that when we look at meta-analysis of pre and probiotics for depression, anxiety, and stress-related behaviors, we're beginning to see more and more that there is a positive effect. But we need to be careful about lumping. Generic term I use, we just call probiotic a probiotic. When in most cases that it'll come down to the specific strain that's been used. And again, understanding why some strains have beneficial effects and others not is going to be an important factor moving forward. This is quite interesting. This is a recent study championed here by Jerry Maloney and the team, where we wanted to look at the same biflongum in individuals that were going through exam stress. And when they went through exam stress, they had changes in their sleep patterns, and we used the Pittsburgh sleep inventory, and we were able to show if they'd taken this biflongum that there was a better sleep quality overall. And so this is something that we're really intrigued about, the relationship between the microbiome, sleep patterns, and also circadian biology. And we have ongoing research now looking at circadian effects, and this is reviewed recently by Paramisa Sen and Gabriel Tofani in the group, looking at the relationship between the microbiome and circadian rhythms, but now here also in relation to the processes underpinning sleep, whether it's neuroinflammation or the blood-brain barrier, the serotonergic system, mitochondrial stress, gut permeability, or the serotonergic system. So stay tuned for more on this. And these are a small study, but it was really pointing us in that direction. And in tandem with the rise of the microbiome as being a key player in relation to brain function, we've seen a whole sea change in our appreciation of the impact that nutrition can have in relation to psychiatry. And the whole discipline of nutritional psychiatry is emerging, and we're beginning to really appreciate this more and more, that we can basically target mental health by targeting what we eat. But the missing part of the puzzle, and this is work that Kirsten Birding-Harold, a postdoc and a nutritionist in my team, is working on, is really triangulating the relationship between diet and the brain with diet and the microbiome and the brain. Because we know diet can have effects directly on the brain, but could many of the beneficial effects of diet on the brain be mediated through the microbiome? And of course, this is work that Carlos and others at the Champilamo are very interested in, using model organisms to tease apart. We're using the human in our studies in terms of creating a psychobiotic diet that might help us in this triangulation. And so stay tuned for that work as it emerges moving forward. In support of that work Kirsten has been doing together with Katrina Long-Smith in the group, is we've given healthy volunteers a specific dietary fiber that we know will influence the composition of the microbiome, and we do see positive effects on this cognitive touchscreen battery, the CANTAB battery, most notably in terms of intra and extra dimensional set shifting, so looking at cortical mediated effects, as well as rapid visual information processing, and we can see very clear beneficial effects. Subtle, these are healthy people, this is not in dementia, so this is an important small study to showcase that we can actually target the microbiome through fiber, because fiber is king in this regard, and we can actually have beneficial effects on brain and behavior in humans overall. And another aspect of what we're doing I want to highlight is work that the wonderful bioinformatician in my team, Thomas Bastianson, has been really developing, and that's looking at 2 time points within the microbiome and looking at changes within that. And so here he paired up with Anand Gaurajan, a former postdoc in the lab, on a chronic social defeat study in animals, and was able to show that the animals that had the biggest shift in their microbiome, a term known as volatility, that they were the most stress-sensitive animals overall, and that there was a correlation, and we used a discovery cohort of mice as well as a validation cohort here to really show that the bigger the change in the microbiome, the bigger the change in the stress response. And that's in mice. What we also wanted to show, well, what would happen in our students as they go through the exam stress period. And again, we were able to correlate the perceived stress score with this volatility index. And so that's really interesting. And at a biological level, we looked at the HPA axis and was able to correlate the corticosterone response in the evening with the level of volatility, as well as the cortical awakening response in humans, which is a really good index of how the hypothalamic pituitary adrenal axis is working overall. So this is something that we've just recently published and is introducing to the field, and we'd love more people to see what are the limitations on the on volatility and the constraints on it as a concept to understand the impact of stress, nutrition, or other interventions have on the microbiome per se overall. And moreover, what we're beginning to see is that when we're looking at stress and depression, that we're beginning to see that it is an unholy trinity. And we had St. Patrick's Day here in Ireland last week, so that's why I'm very much in my trinity mode. But you know, that depression is made up of deficits at the level of stress, immunity, and the microbiome, and that if we want to fix it by a treatment, then we need to target all three, and that's going to be really important moving forward. I also just want to give a shout out that while in the gut feelings approach, most of the behaviors that we're interested in have been focused on psychiatry, we're also beginning to see that in neurology, there's a really growing interest in the role of the microbiome and the gut in general in shaping brain and behavior. We're seeing this I've already mentioned in autism, but also in neurodegenerative diseases like Alzheimer's and Parkinson's, where we're now beginning to see a gut first aspect in ALS and Huntington's disease, in stroke and brain injury, and multiple sclerosis. It's become very well characterized, and epilepsy. And epilepsy is one of these diseases that has been managed in certain cases by diet, using the ketogenic diet, and work from Elaine Sau's group has shown that this is mediated, at least in animals, in part due to its effects on the microbiome. So, you know, understanding diet, microbiome, gut-brain axis function is going to be really important in all aspects of brain and behavior. And so we're getting to the stage where we can't have a neuroscience conference without having a microbiome section to it overall. There are challenges, and I just wanted to highlight some of them. The main challenge is really we don't know what a normal microbiome is. There have been some attempts to do that, most recently in the Dutch cohorts, to try and understand it. But you can go to your doctor and get your blood pressure measured, your cholesterol measured, and they can see where you are with the normal microbiome. You go in within the normal levels, but you go in with a microbiome readout, and all they will do is look at you in despair and wonder what to do with this. We're seeing changes with citizen science and innovative platforms with longitudinal measures and sequencing costs coming down. We have challenges around sample size, because the sample size, whatever you choose in human cohort studies, is always wrong. And it's like GWAS was 20 years, we're getting more and more increase in this. And we're getting newer bioinformatic tools which can really tease out what's going on. We're seeing that personalized diets and interventions are really important, and that interactions with host genetics, the diet and environment are going to be really important. We're beginning to see ways that not just the bacteriome, but also other components, and particularly phage and fungi, are really playing a role. And there's becoming a greater appreciation of the wonderful utility of model organisms, whether it's C. elegans, Drosophila, or zebrafish, in teasing apart some of these at a scale that you just cannot do in rodents or in humans overall, and for looking at some of the evolutionary aspects of this. How do microbe signal to the brain is still a really big question, and we really need to get at mechanisms. There's a lot of innovation here. I just threw in two of them that are really important. One is the development of smart pills. These are sensors that can go into the gut and send signals back to what has actually been going on in parts of the intestine, because reliance on fecal material may not always be appropriate. And then there's the whole magic toilet aspects, which I always feel will go down a dream in Japan. But this is innovation coming out of Stanford and other places, where they're going to have a mass spectrometer attached to your toilet and measure what's going on overall. We need to really understand what are the molecular targets of these microbial metabolites and how these are working, and then the barriers. Both the gut barrier and the blood-brain barrier are going to be really important as we move forward. But the good news, these are all hurdles and challenges and excitement, and we're going to see a lot more in it. The good news is that unlike your genome, which all you can really do is blame your parents or your grandparents, your microbiome is potentially modifiable. And so that's really an important aspect, and understanding what the constraints and that modifiableness is going to be really important moving forward overall. So a big shout out to our brilliant team who have navigated this crazy year of COVID overall so well, and remain productive and keeping so many shows on the road. And stay tuned for many exciting work, all of our funders and collaborators. And I'm back to Hippocrates, who's also thought to have said, and again it could be fake news, that let food be thy medicine. And we'll change that to let food for your microbes be medicine for your mind. And just remember, going back to what I said at the very beginning, that sometimes it's good to turn things upside down and you can see them in a different perspective. So thank you very much for listening today, and I'm more than happy to take questions, and I will stay on afterwards to chat with anyone who is so inclined. And again, thanks to everyone at the Champilimo and everyone involved in this series, Carlos and Asea, and everyone for inviting me to be part of such a great seminar series.
Thank you so much. So this was an amazing talk. Let's go to the questions. Okay, so the first one. Great talk. Can microbiota be targeted and objectively assessed for meaningful interventions? Are there clinical trials ongoing for neuropsychiatric conditions such as ASD, especially as this area seems to be targeted by pseudoscience?
That's a wonderful question. I often put it in my talks about that very question. There is a lot of snake oil in this field, and we need to be really careful about what we're trying to always ask for evidence. And the questioner is asking, where is the evidence right now? There hasn't been so much longitudinal data so far in ASD or ADHD or otherwise, but it's now being looked at. We're seeing big initiatives coming forward from Wellcome and from other funders who want to do this. There is some work going on as part of cohort studies. Thin Brain, for example, in Turku in Finland are doing this. Also work coming out of North Carolina, and the Human Connectome Project are looking at this in large cohorts, and then of course some of them will develop ASD or other neurodevelopmental disorders. So I think that's where we're going to see more and more of this. It needs to be done properly, and we need to get more evidence so that we can put an end to pseudoscience and really become evidence-based.
If a cesarean has such dramatic effects on the gut bacteria, and there is this strong gut bacteria brain link, wouldn't one expect a dramatic effect of cesareans on brain function and incidence of neurological and neuropsychiatric disorders?
That's a great question, and it was one of these things that we were interested in doing. So first of all, this is within a mouse system where their microbiome is only influenced, is very much controlled. So the correlation of that to humans, we don't know. I mean, there is some epidemiology data, but most of it doesn't point to something so severe overall. We did some studies in, I was going to say normal, but Irish medical students, and we stratified them based on their mode of delivery. And we were able to show that those that were born by C-section had a greater stress response in the lab, and also a greater stress response to exams. Now, again, there are many other factors at play, and it mightn't just be microbiome, but it is one of these things. You will be surprised, though, that there isn't really good quality data out there as much as we would like, and overall what's driving it. What we're beginning to see in the epidemiology data is it's maybe not the C-section, but what's driving the C-section that might be one of the risk factors overall.
Since humans and mice have different microbiota, there will be different interactions with a new organism. Could it be that the different hosts could require a different bacterial strain as a probiotic?
Absolutely, and I think it's a really good question. And these types of species-specific effects are going to be really important. Also, the geography and biogeography of the rodent gut is going to be very different than that of the human. So these are really important aspects, and you would almost be surprised that anything would translate from an animal to a human, but it does. So that also tells us that evolutionary, there is a lot of conservative conservation going on as well.
With respect to diet changes for the adolescent brain, what was the reason for focusing
so vitamin
on
A is interesting.
A?
So this was work that they were the group in Florence were already looking at vitamin A and polyunsaturated fatty acids, and there's a lot of data of direct effects of retinol on brain function. So that they weren't assuming that it was going to have, and we don't know whether it was the vitamin A alone or the vitamin A coupled with the polyunsaturated fatty acids. And I think it's probably the polyunsaturated fatty acids that were having more of the effect than the vitamin A, but we can't rule that out overall. And so they just chose two components that have been shown by other groups, including groups in Bordeaux, to have positive effects on cognitive function, especially in animals that have been given obesogenic diet.
Can you comment on interplay between the host immune system and microbiome in some of the phenotypes you've described, particularly for interventions that affect early life development, C-section, early life antibiotics, gen-free mice? Might neural phenotypes in part be due to altered immune function indirectly modulated by gut microbiome?
So how much time have you got? I mean, this is a wonderful question, and it's a really important question, and it's something that we're really, really focusing on. So I have a wonderful Portuguese PhD student, Joana Pereira, and my team who's working on this in both early life and in aging to try and tease apart what is the role of the immune system in mediating this. And I really think that systemic immune system and how and also monocyte traffic into the brain is going to be really important in mediating some of these effects. It's hard to disentangle about which comes first. And there are a multitude of different mechanisms at play here. So I probably didn't play up the host immune system enough, but it's really important, both in terms of acute effects and also the adaptive immune system.
Thank you for a great talk. I heard of ketogenic diet being proposed as an auxiliary treatment for bipolar disorder. What is it about this diet that seems to help here and in epilepsy, as you mentioned? Yeah,
so there is work coming from Zoltan Zarnai's group and James Cook University in Australia and others in animal models of bipolar and schizophrenia and the work that Elaine Sau's work has really focused on in UCLA in animal models, showing that it is due to its impact on the microbiome and how that microbiome changes the glutamate GABA balance in the brain. So it's basically down to perhaps glutamine and metabolites of the ketogenic diet that are having a positive effects there. So that's one of the things that we're beginning to explore more. It's quite a busy area right now. A lot of people are focused on how much of the beneficial effects of the ketogenic diet are mediated through the microbiome.
Are there specific peripheral molecular biomarkers, short-chain fatty acids, bacterial metabolites, and or signaling molecules that can be used as a routine screening for changes of microbiota of patient with respect to his normal baseline that can signify disease? Thank you.
Great question. We're beginning to understand. There's been some really exciting work on looking at the blood metabolome, and that it is a good surrogate marker for the diversity of what's going on in the microbiome. So metabolites, in principle, could be used. Work from Peter Dorstein's and Rob Knight's group are showing this more and more, but like it hasn't made its way into routine clinical diagnostic practice yet. So I think that's, we need to see a little bit more details on that. But it reinforces that it's not, when we're talking about the microbiome, we get very obsessed about a compendium of what's there. But what's there isn't as important as what we're doing. And that's where the metabolites come in. And what they're doing is going to be crucial in this regard.
All right, last couple of questions. What effects does Enoline have? Enoline, I don't know if it means insulin, at the microbiome level.
Yeah, so inulin is a prebiotic, and so it stimulates a lot of these short chain fatty acids and has a multitude of effects overall. So that's one of its main aspects overall.
And last one, are volatile fatty acids also crucial culprits like short chain fatty acids? What other potential metabolites do you suspect could possess the key to understanding the axis?
Yeah, I think we need a lot more metabolic insight. So there's some really nice work being done on TMAO and its role, and then tryptophan metabolites. We're very interested in the tryptophan metabolites and their ability to indoles, to bind aryl hydrocarbon receptors, and how that could be mediating some of what's going on. So and bile acids. So if I was thinking of three, other ways, it would be like TMAO, bile acids, and tryptophan metabolites. But there's a lot. We're only scratching the surface. We're only really scratching the surface.
All right. Well, thank you so much, and thank you everyone who joined us today. Our next speaker will be Daniela Carnaval next Monday, talking about neuroimmune interaction in cardiovascular diseases. So for those of you who signed for the Meetup the Speaker session, I'll see you now in the Zoom room. I already sent you all the link, I think. And for everyone else, I hope you all have a wonderful week.
Wonderful.
Bye.